Oxygen is vital for the energy-releasing process of aerobic respiration, and animals have evolved a wealth of different mechanisms for extracting oxygen from their surrounding environment. Water-breathing animals face a more difficult task than air breathers, since water contains less oxygen than air. It's also denser and more viscous, so a greater amount of energy must be expended in moving water across the respiratory surface where gas exchange takes place. By contrast, eliminating waste gases, such as carbon dioxide, may be simpler because carbon dioxide is freely soluble in water, and so doesn't accumulate in the blood of water breathers. In this program, we should be looking at two organisms which face different respiratory challenges. One is the air-breathing bird, with its high demand for oxygen associated with sustained flight. The other is the shore crab, which may lead a more sedentary existence, but often encounters low oxygen levels. Apart from the buffeting it receives from the waves, a shore crab can often be stranded on the beach whenever the tide ebbs. To avoid water loss in the summer heat, it will often retreat into rock pools. Under these conditions, it may experience short periods of relative oxygen lack. How does the animal cope with these reduced oxygen levels? One clue comes from a characteristic behavior pattern that's often seen at low tide. It consists of sitting at the water's surface with the anterior part of the body raised and bubbling air through the gills. In order to understand the significance and underlying mechanism of this behavior, called the immersion response, we need to look first at the anatomy of the gills and at how ventilation takes place. Beneath the hard outer skeleton of the crab lie two sets of gills, hidden and protected in the branchial chambers. The detailed structure of each gill is best seen in a scanning electron micrograph, which reveals a number of regularly stacked lamellae that greatly increase the surface area available for gas exchange. A transmission electron micrograph through a single lamella shows a relatively thick lining layer of chitin, which confers rigidity and some degree of resistance to collapse in air. Water is circulated within the branchial chambers by the pumping action of a specialized appendage called the scophognathite, one above each set of gills. The beating action of the scophognathite causes water to be drawn in near the base of the legs and to be moved upwards and forwards over the gills before being propelled out over the mouth. Here we see the blade of the scophognathite oscillating in the narrow channel at the front of each branchial chamber. If we place blue dye near a living crab, looked at from above, the current is drawn in, passes over the gills, and we can see it here ejected forward in a powerful stream. In a semi-transparent prawn, it's easier to see the scophognathite beating at the front of the branchial chamber, and its beating draws water forward. Dr. Ted Taylor of Birmingham University has worked on respiration in crabs and explains how it can be studied experimentally.
We can record the small hydrostatic pressure variations generated in the branchial chambers of the crab by the scophognathite when it's ventilating the gills, and by using a sensitive pressure transducer like this one here. This is recorded via a thin plastic tubing to the crab, and this tube is inserted below here, near the base of the pincers, so that it records the pressure generated by the scophognathite, which lies here. In this crab, as well as inserting this tube to measure branchial chamber pressures, we've inserted a pair of fine wires above the dorsal heart to pick up electrical activity associated with the heartbeat, in other words, the electrocardiogram, and this gives us a readout of heart rate. Both these traces are registered on this pen recorder here, which is running and recording activity from our crab. The top trace is the ECG, and each one of these blips on the trace here is a beat of the heart. So you can see it beating away there. The bottom trace is a second time marker, and you can see that the heart rate is roughly 1 a second, slightly slower. This fluctuating recording here is, in fact, the pressure, the water pressure generated, hydrostatic pressure generated in the branchial chambers by the scophognathite, recorded from the pressure transducer. If we were to look at what ambient pressure was, in other words, the water pressure above the animal is here. In other words, our animal, the branchial chambers are working at a pressure below ambient because the scophognathite has a sucking action. It sucks water in round the bases of the legs and over the gills. For that reason, the pressure that we measure is subambient. If we watch this trace for a while, then the crab may do something rather interesting. And there it was. For a short period, the pressure became above ambient. This is because the scophognathite is no longer sucking water from around the leg bases, over the gills and out round the mouth, as in normal flow. Instead, it's blowing the water backwards from around the mouth to emerge round the leg bases. This infrequent and brief reversal of flow probably serves to clear detritus from the gills and is therefore analogous to a cough when the animal is submerged in water. It has a more interesting function when the animal is in reduced oxygen partial pressures. Now let's see what happens to these pressure fluctuations in the branchial chamber and to heart rate when we reduce the oxygen partial pressure in the water surrounding the crab. What's happened here is the animal has shown a reversal that you should recognise from before, and at the same time the heart rate suddenly increased. Instead of the pressure here, however, returning back to the normal pattern, we're getting these rather jagged and peculiar positive pressures in the branchial chamber, in other words above ambient, and these are associated with the bubbling activity of the typical immersion response.
In oxygen-depleted water, the crab emerges at the air-water interface, and instead of sucking water in around the leg bases and over the gills, it draws air in at the mouth and blows it over the gills to emerge around the leg bases. This gives the characteristic air bubbling effect of the so-called immersion response. The bubbling of air through the branchial chambers during immersion helps to aerate the water surrounding the gills. The sudden rises in heart rate increase the rate of delivery of oxygen to the tissues.
In order to understand the respiratory significance of the immersion response and the associated air bubbling to the crab, we need really to go inside the animal and to look at what's happening to delivery of oxygen to the tissues by its blood, or hemolymph as it's called in this case. By injecting dye at the base of the gills, we can trace the path of the hemolymph through the lamellae, and it's here that gas exchange takes place. The animal's hemolymph contains a blue respiratory pigment called hemocyanin. It's possible to sample the hemolymph as it leaves the gills and measure how much oxygen it's carrying at different oxygen partial pressures. Here are the results for 3 possible cases. High oxygen partial pressures, for instance when the crab is submerged in fully aerated seawater. Low oxygen partial pressures, as when the crab is in an oxygen-depleted rock pool. And intermediate partial pressures found in the gill chamber when the crab is performing its immersion response and bubbling at the air-water interface. Although the oxygen content of the blood pigment increases with higher oxygen partial pressures, the results don't fall on a straight line. To understand this non-linear relationship, we need to build up a full profile of how much oxygen the hemocyanin is carrying at different oxygen partial pressures. This is called the oxygen dissociation curve. We can determine the oxygen dissociation curve for the crab's blood quite simply by taking advantage of the fact that it's blue when oxygenated and then goes colourless as we remove oxygen. All we have to do is insert it into a simple colorimeter like this, which measures the degree of blueness in the blood, and then connect it by a piece of simple rubber plumbing to a vacuum pump like this one here. When we switch the pump on, it will draw a column of mercury up here, as you can see, which is merely registering, on a simple meter rule, the reduction in pressure below the barometric pressure induced by the vacuum pump. Now, in order to determine the dissociation curve, what we do is do up this pinch tap on the side tube and take the system away from the pump, which we can now switch off. And then by progressively undoing this tap and doing it up again, we can introduce that much gas, air, back into the system. In other words, the volume of this piece of tubing, stage by stage. And by doing this, we get an indication of the increase in blueness of the blood related to the change in the pressure on this mercury column. By doing the calculations necessary, we can elaborate a dissociation curve. The complete oxygen dissociation curve for crab hemocyanin looks like this, and its S shape reflects special properties of the blood pigment. The 3 points plotted earlier all fall on this curve. The oxygen partial pressures at which the crab shows its immersion response fall within the steep part of the curve. Here, a small increase in oxygen partial pressure gives a large increase in the amount of oxygen combined with the hemocyanin.
Another problem to contend with is the disposal of carbon dioxide, which can cause the acidity of the blood to rise if it's allowed to accumulate. Carbon dioxide dissolves readily in water, and since the crab's gills are still in contact with water during immersion, the gas can be eliminated. But it's a very different picture for crabs stranded in air between tides. The gills collapse and can no longer function adequately in gas exchange. So oxygen levels fall while carbon dioxide levels rise. This causes a drop in pH from 7.84 to 7.71, a small decrease, but one of great significance to the animal. How does this affect the delivery of oxygen to the tissues? The secret lies in the effect of acidity on the oxygen-carrying properties of hemocyanin. When the crab is submerged in fully aerated water, the oxygen dissociation curve of the pigment looks like this. When the crab is in air, the acidity of the hemolymph increases due to dissolved carbon dioxide. The curve becomes shifted to the right because the affinity of the pigment for oxygen falls. This is known as the Bohr shift. It means that for any oxygen partial pressure in the blood, the oxygen affinity of the pigment is lower in air than it is in water. How does this affect the amount of oxygen delivered to the tissues? For animals submerged in water, the oxygen content of the hemocyanin varies from near saturation around the gills to about half saturation in the tissues. So, the amount of oxygen that can be delivered to the tissues is shown by this bar. For animals in air, the oxygen content of the hemolymph varies from this value near the gills to this near the tissues. Since this falls within a steep part of the curve, the amount of oxygen that can be delivered to the tissues is as much as, or greater than, that in water. So, changes in the oxygen-carrying properties of the hemocyanin do occur under different environmental conditions. This allows crabs to be highly efficient at extracting oxygen from air, oxygen-depleted water, and from water with normal oxygen content. In vertebrate animals, we see different mechanisms. The respiratory system of birds is a particularly striking solution to the problem of attaining sufficient oxygen. A bird in flight expends more energy, weight for weight, than a mammal walking or running on the ground. Furthermore, a bird's respiratory system can deliver enough oxygen for it to fly at high altitudes where a mammal can barely function. What type of respiratory system can meet these demands? Professor Pat Butler of Birmingham University.
The respiratory system in birds is quite different from that in mammals. These are two casts of the respiratory system in the pigeon, and there are two lungs here just beneath the rib cage, and this really is about all that the respiratory system in birds has in common with that in mammals, because attached to these lungs is quite an extensive system of air sacs which occupy a major volume of the body cavities. Now, these are the bones in the animal, and if these are dissolved away, it's possible to see that the air system also extends into these bones. So this is the cavity inside the bone of the forebone of the wing. This is the air cavity in the sternum, and this is the air cavity along the spinal cord. The air sacs themselves are very thin-walled and very poorly vascularized, so they themselves are not involved in gas exchange. It's just the lungs that are concerned with the process of gas exchange, and the anatomy of the lungs can probably best be seen in a diagram. Each lung is surrounded by a group of air sacs, some at the anterior end and some at the posterior end. As mentioned earlier, these hollow sacs aren't involved in gas exchange, but being thin-walled and elastic, they can easily change volume, expanding and contracting to ventilate the lungs like a set of bellows. The entire respiratory tract is an intricate system of air passages. Inhaled air passes down the trachea into each lung via a large tube called the primary bronchus. This then divides into smaller secondary bronchi, some at the anterior end, called ventrobronchi, and others at the posterior end, called dorsal bronchi. The secondary bronchi are joined together by numerous interconnected tertiary bronchi that make up the bulk of the lung. These fine tubes are the sites for gas exchange. Looking at a three-dimensional diagram of the tertiary bronchi, we can see that they are hexagonal in shape, and a cross-sectional slice through the bronchi shows this very clearly. This scanning electron micrograph picks out the hexagonal nature of these tiny air passages. Alternatively, we can cut a longitudinal section down the length of the tertiary bronchi. This reveals an intricate system of pit-like depressions in their walls. These depressions lead to small openings and then to tiny air capillaries. The air capillaries entwine closely with the blood capillaries. So as the blood passes through each tertiary bronchus, it undergoes gas exchange, gaining oxygen and losing carbon dioxide. An electron micrograph shows nicely the close relationship between the blood capillaries and the surrounding air capillaries. The distance between the two is no more than about 1/10 of a micrometer, a very small barrier to diffusion between the air and the blood. Since the tertiary bronchi within the body of the lung are open at both ends, air can theoretically flow through them in two directions, from the anterior to the posterior, or vice versa. In fact, flow detectors, implanted in the respiratory tract, reveal an extraordinary result. Gas flows from the posterior end of the tertiary bronchi to the anterior, both during inspiration and during expiration. So how is this unidirectional flow achieved without any evidence for physical valves to guide the passage of air? Now, probably the most plausible explanation for this that we have at the moment is based upon the shape and the angles of insertion of these tubes themselves. And if we look at this model here, this cast of a bird lung, now in this model, most of the tertiary bronchi have been removed so that we can see the primary bronchus and the posterior secondary bronchi. It looks like the geometry of the air passages, their relative width, and the momentum of the air itself are sufficient to guide the passage of air in the lung. In other words, the valving is aerodynamic in nature. So during inspiration, for instance, the density and velocity of the incoming gas are such that air tends to flow down the main bronchus rather than turn the acute corner into the ventrobronchi. A decrease in the diameter of the primary bronchus at the branch point could improve this valving by increasing the velocity of the incoming gas. During expiration, how can we explain the airflow into the dorsal bronchi, but its absence in the midsection of the primary bronchus? In this case, anatomical features may be important. For instance, the dorsal bronchi are well aligned to receive the jet flow of air direct from the posterior air sacs, with relatively little passing down the main region of the primary bronchus. By contrast, air leaving the anterior air sacs must turn a sharp corner from the ventrobronchi into the primary bronchus. This change in direction is probably achieved by a region of high pressure in the midsection of the main bronchus. This would in turn oppose any flow from the posterior air sacs along the primary bronchus. Although this explanation is still somewhat speculative, it's already clear that with this pattern of airflow, there's little mixing of inhaled and exhaled air. This minimizes the amount of dead space air within the lungs containing relatively low amounts of oxygen and high amounts of carbon dioxide. But another factor which affects the efficiency of gas exchange is the relative direction of blood flow and airflow. We know that air flows in the tertiary bronchi from the posterior end of the lung to the anterior. The surrounding blood vessels seem to run across the tertiary bronchi. So the direction of blood flow is at right angles to the direction of airflow, a cross-current system. This arrangement makes the exchange of gases more effective than in the mammalian lung, particularly at high altitude.
How can we link the respiratory system of birds to their physiological needs during flight at any given altitude? Wind tunnel experiments have given some idea of the oxygen uptake that occurs during sustained flight. But the birds show an abnormal flight pattern, and so results have to be treated with caution.
Respiratory frequency about 15 a minute before takeoff.
15 breaths a minute, yeah.
But Professor Butler and his colleagues have demonstrated that physiological data can be obtained from free-flying birds. Tiny transmitters can be implanted in the animal to measure heart rate, respiratory frequency, and eventually even oxygen uptake during natural flight. Results show that the oxygen uptake of birds during flight is over twice that of mammals of a similar size running at their maximum sustainable speed. But interestingly, flying mammals, such as bats, consume oxygen at similar rates to flying birds. So the increased oxygen uptake during flight isn't dependent on an avian type of lung. However, even flying mammals, such as bats, aren't capable of the sustained flight at high altitude known in birds. The most dramatic example is the bar-headed goose, which has been known to migrate at a height of about 9,000 meters across the Himalayas. The oxygen partial pressure at these altitudes may drop to a third of that at sea level. How do the mammalian and avian respiratory systems compare under these conditions? The oxygen partial pressure of air inhaled at sea level is shown by this bar. In a bird, the arterial blood leaving the lung can achieve an oxygen partial pressure about 60% of this value. This level of effectiveness is certainly matched by the human lung. The picture is very different at high altitude. At the top of Mount Everest, more than 8,500 meters up, the partial pressure of oxygen in inspired air drops to about a third of that at sea level. After acclimatization, a human can almost bring the level of oxygen in the blood up to that of the air breathed in, just enough to reach the peak without supplementary oxygen. By contrast, the bar-headed goose can be exposed to simulated altitudes of over 10,500 meters. Although the available oxygen drops to less than a fifth that at sea level, the bird can still transfer fractionally more oxygen into its blood than a human can at a lower altitude. So the limits of an animal's respiratory system may be most rigorously tested under conditions of strenuous exercise or poor oxygen availability. And it's precisely under the conditions of prolonged flight at high altitudes with little oxygen that the respiratory system of birds comes fully into its own.